In December of 2014, computational biologist Lear Pachter published a blog post that sent ripples through the scientific and public communities. Titled, "The Perfect Human is Puerto Rican," the post detailed a thought experiment that involved constructing a hypothetical perfect human genotype based on available genetic data. While Pachter himself characterized the experiment as a tongue-in-cheek exploration, the implications and reactions to his findings were far from trivial, as many were taking it as a serious scientific study. What many people failed to realize was that Pachter was just trying to prove a point. But what point was he trying to prove? Pachter conducted the experiment as a response to famed American molecular biologist and Nobel Prize winner James Watson. Watson's contributions to science, particularly his role in uncovering the structure of DNA, are groundbreaking and have had a profound impact on genetics and molecular biology. However, his career has been marred by controversial statements and views, especially regarding race and intelligence. In various interviews and writings, Watson has expressed views suggesting that genetic differences contribute to variations in intelligence among different racial groups. In fact, one of his obsessions has been to improve the imperfect human via human germline engineering. This is disturbing, especially for the fact that for years, Watson presided over Cold Spring Harbor Laboratory, which actually has a history as a center for eugenics. It was in this context and his own experiences with Watson that inspired Lear Pachter to conduct his own experiment using the structure of DNA. Pachter's starting point was a database of genetic information specifically focusing on single nucleotide polymorphisms, or SNPs. These are variations in a single DNA nucleotide that occur at a specific location in the genome. The database contained information on SNPs associated with a variety of traits, some of which were considered positive or beneficial. Using this data, Pachter created a hypothetical genotype by selecting the best allele or variant for each SNP associated with a desired trait. This constructed genotype represented an idealized individual with a combination of advantageous genetic variations. The next step involved comparing this perfect genotype to existing genetic data. Pachter used data from the 1000 Genomes Project, a massive international research effort to map human genetic variation. By performing principal component analysis, a statistical technique used to identify patterns in data, he found that the perfect genotype clustered most closely with individuals from Puerto Rico. Pachter's declaration that the perfect human is Puerto Rican was met with a mix of amusement, skepticism, and outrage. It's important to emphasize that this conclusion was based on a highly simplified and idealized model of human genetics. The experiment was based on a limited set of SNPs associated with specific traits. The complex interplay of genes and environmental factors in determining human phenotypes is far more intricate. In fact, Lear Pachter's limited experiment was not to proclaim a perfect human, but to demonstrate how absurd Watson's ideas about race were, because even in this limited experiment, the results should have been Watson's ideal type, but they weren't. Lear Pachter's experiment, while interesting, ultimately falls short as a serious scientific endeavor. However, it serves as a cautionary tale about the limitations of reducing human complexity to a single idealized genotype. By understanding the complexities of genetics and the importance of human diversity, we can move towards a future where genetic information is used responsibly and ethically for the benefit of all. Let us know what you think in the comment section below. Help us to spread this video by sharing. Don't forget to subscribe and turn on the notification bell for more videos.